All right, so we're studying enzyme kinetics now, and enzyme kinetics are so important because they determine the mechanism of any given reaction. They can determine the effects of regulatory molecules. They can show the impact of changes on reaction conditions. There's so many different things that enzyme kinetics can tell us. And so when we talk about it in terms of equating free energy and equilibrium. And so the different things that we can measure are how fast a reaction goes, which is the reaction rate or the velocity of a reaction, so the speed of a reaction. And we measure that in concentration of product versus time. We can also measure the substrate concentration, how that affects the rate of a reaction. And then every single reaction has its own specific rate constant of that reaction. And so we can calculate the velocity of a reaction if we know the rate constant and we know the concentration of the substrate. And this is only true for first order reactions, because first order reactions depend on the concentration of the substrate at the initial part of the reaction. That's what the little I is for. So when we look at this, our rate constant units are seconds to the -1. And our velocity units, therefore, are dependent upon the substrate, and the substrate concentration tends to be in molarity or millimolar or something like that. And then, so velocity will be the concentration of the substrate times the reaction constant. So that'll be molarity times seconds to the -1. And so when we talk about our reaction, this is kind of what we had before, where we had an enzyme that we were adding a substrate to. And so we made enzyme substrate. And remember, when we did this before, we made enzyme product, and then we had enzyme plus product. But really, once that product is made, it gets kicked out so fast that we're not really worried about that rate being dependent upon anything. It just happens so insanely fast that we don't need to worry about it. So as soon as the product's made, it's released. So that's why we go from ES to EP, to E plus P, okay? So each of these different stages has a different rate constant. So if you're going in the forward direction from enzyme plus substrate to enzyme substrate, that's K1. If you go from enzyme substrate to enzyme plus product, that's K2. Now, we typically never see you going from enzyme product back to enzyme substrate. So you don't have a reverse arrow here, but you can go from enzyme substrate and then the enzyme just lets go of the substrate instead of turning it into a product. That happens. So that's our K to the -1. So just kind of keep that in mind as we go through our different examples. So when we talk about enzyme kinetics, typically we talk about Michaelis-Menten kinetics, and this is for first order reactions. And we always do this where our substrate concentration is much, 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 much greater than the enzyme concentration. And so this allows us to understand what's happening with the reaction. Now, I want to talk about how we get the data in B. So this is really kind of like figure A down here. So if we take a graph of concentration of product versus time, what is going to affect how fast product forms, right, is the concentration of substrate. So if we have a low concentration of substrate, it takes quite a long time to make product. But if we have a high concentration of substrate, ooh, that goes really fast. But what you'll notice is that it takes, that you see it less of a change the higher you get in substrate concentration. So we know the velocity, and here we're measuring the velocity. If you get the slope of that line, that's our velocity of our reaction. And so our velocity can vary with substrate concentration, but as we get closer and closer and closer and closer to some set substrate concentration, you're not going to be able to go any faster. And so when that substrate concentration gets high enough, we can say that we've reached the maximum velocity of a reaction. And so what we've done is this line right here, we take the slope of that, and here's our data point. Then we take the slope of this one, and that's the data point, and so on and so on and so on. And so what you're going to see is that there's this maximum velocity that this reaction is trying to get to. It's never quite going to get there, but this right here is our Vmax, right? That's our Vmax line. And so from here, we can determine the mechanism of the reaction. So we know that at our Vmax, like as we're approaching our Vmax, our substrate is binding and binding and binding to our enzyme so much that our binding sites of our enzyme are completely saturated with substrate. So we know that we can infer some things about our enzyme, knowing that all of those binding sites are full at Vmax. And we're going to apply that. There's a fantastic animation that talks about enzyme kinetics that you should go and watch. All right. So when we do all of these Michaelis-Menten calculations, you're always going to be doing this in steady state conditions, because for any given reaction right here in the beginning, we have some pre-steady state conditions that are happening. And so things aren't quite going to be constant. And so everything that we do is in this steady state condition where our enzyme concentration isn't really changing. Free enzyme concentration are free. Our enzyme bound to substrate concentration isn't really changing, but we do see a decrease in substrate and an increase in product. And so when we do this, what we have to see is what kind of information can we pull out of this? And so what we're going to do is we're going to plot these steady state conditions, just like we did before, where we graphed our initial velocity versus our substrate concentration. And so what we find is that we can get a Vmax, right? This is our Vmax, what everything's trying to get to, Vmax. It's the maximum velocity of that substance, of that enzyme. And then if we take the half Vmax, so this is 1/2 Vmax, and we dash that out until it hits our curve, and we come down, we're going to get a substrate concentration. And that's the substrate concentration where you've reached half of the maximum velocity. And we call that the Km, or the Michaelis-Menten constant. And the Michaelis-Menten constant is actually a concentration, a measure of the concentration of the substrate, right? And so the Km really tells us about catalytic activity. If you have a really low Km, a small Km, that's telling you that you only needed a little bit of substrate in order to get maximum velocity of your reaction. And so a low Km tells us a high catalytic activity. So we can use that. So let's come and use this equation that we had before. And so what is really Km? If you went through and mathematically calculated Km, what it really is K to the -1. So that's the enzyme substrate breaking down into enzyme plus substrate, plus the rate of going from enzyme substrate to enzyme product, right? So really what these two factors here, this is really breakdown of our enzyme substrate complex for any given reason, whereas K1 is the only one that is formation. So Km is really binding affinity, right? Oh, I already wrote it. Look at this. It's Km, breakdown over formation. So Km is really, really binding affinity. And then you can also mathematically calculate the velocity of the reaction. And so we're going to come back to that formula in just a few minutes. So if we look at a typical Michaelis-Menten enzyme, okay, so if we look at the Vmax of any given reaction, this is one enzyme, and the thing that we're changing is the red is a high enzyme concentration, the green is a low enzyme concentration. If we change the concentration of the enzyme, the Km will not change. It is constant. Michaelis-Menten constant. It's a constant. No matter how fast that reaction is, right? So if it goes all the way up here, this is our Vmax, and we go half down here, here's our half, come down, we're going to get 50 micromolar. Whereas if we have a low concentration, here's our Vmax, half of 20 is 10, come down, you're still at 50 micromolar. So Km is a constant. But what we do see change is Vmax. So as we increase our enzyme concentration, the Vmax, that max velocity of our reaction, is going to increase. But our binding affinity will not, because it's a constant. That's why we call it the Michaelis-Menten constant. So sometimes it's nice to look at a graph like this, and sometimes it's nice to be able to look at something a little bit clearer that's a perfect line. And so what we do is we take our equation that I showed you before for our velocity of our reaction, and we do the inverse. So this side of the equation, we take the inverse of it. This side of the equation, we take the inverse of it. And then we rearrange this formula in order to fit that of a normal line. Y is equal to mx plus b. We've all seen that before. And so when you separate out the b from here, right? If we separate it out, our concentration of substrate cancels out. And so we just have 1 over our velocity is equal to Km over Vmax times our substrate concentration plus 1 over Vmax. And so if you kind of look at this, what you can see is how this equation now lines up perfectly with our equation of our line. Y is equal to mx plus b, right? So everything lines up. So what does this tell us, and what can we get from this information, right? So if we plot just what we did before, this is high enzyme concentration, this is low enzyme concentration. What we can easily see is where we intersect with the X and Y axis tells us our Km and our Vmax. Now, the intercept is 1 over Vmax, and this is 1 over negative Km. So you do have to remember that it's kind of swapped, but I'll show you that. But our axes really haven't changed other than being the inverse. This is 1 over substrate concentration. This is 1 over that initial velocity. And so what we can see is that as we change our enzyme concentration, we have one axis that gets changed, but not the other. So here we have a difference in Vmax, and here we have no difference in Km. So it's just an easier way to look at it. And when we talk about inhibitors, I'll show you some nice and easy ways to look at that. So when we talk about enzymes, the other thing that we have to talk about is their catalytic efficiency. And so catalytic efficiency is a combination of two things. It's a combination of the turnover number, so the maximum amount of catalytic activity under saturating levels of substrate. So you have the maximum amount of substrate possible, and the Km. And we know that Km is our binding affinity, right? So that's binding. So how well an enzyme works is dependent upon, once everything's bound, how quickly can it turn into product. That's Kcat, or catalytic efficiency. And then Km is the binding affinity. So it's really the two different things together. So when we talk about the rate limiting step, right, that's Kcat. So you have everything bound maximally as possible. How quickly can you make product? And mathematically, we can calculate that. We can say, okay, what's the Vmax of our reaction divided by our enzyme total? And some of the enzyme is free, and some of the enzyme is bound to substrate. So the total enzyme is those two things combined. So there are lots of enzymes that have the same Kcat value. They can, once they're all bound, they can go just as fast as any other. But they have different catalytic efficiency, which is the combination of the two, because they have different binding affinities, or Michaelis-Menten constants, right? So just remember that catalytic efficiency is two things. It's Kcat, which is our turnover number, divided by our binding affinity, which is Km. And remember your units, Kcat is going to be seconds to the -1, right? It's how fast you can do something. And then that's over molarity. So it's molarity to the -1 times seconds to the -1. So when we talk about enzyme activity, right, we've talked about so many different things, that can affect enzyme activity. And two of the things that we haven't talked about yet are pH and temperature. And we know that there are certain proteins that work at certain pHs. And that really makes sense. Hold on, y'all. See if I can pause this. Maybe. So for example, pepsin works in the stomach. And so we know the stomach's pH is about 1.5, 2, something like that. So we can see that pepsin works really, really well at pH 2, but doesn't work at all at even physiological pH of 7. But glucose 6-phosphatase, this is something that works in the liver. This one is very, very active at pH 7. Arginase is another one that works at a different pH 9.7. So we know that pH has an effect on the chemistry of the active site. It has an effect on really the interaction of the amino acid R groups, so that it would change structure. So if your structure changes, your activity changes, right? The same thing's true about temperature. Most things we know, right? We say, oh, it works really well at body temp, 37 degrees. So E. coli DNA polymerase, absolutely true. Works great at 37 degrees. Taq polymerase, remember, this is the polymerase that we used in PCR. It is home inside of a very, very heat-stable organism, T. aquaticus. And so it has barely any activity at 37 degrees Celsius, but as we go up to like 80 degrees Celsius, and remember, water boils at 100. So this is pretty hot. It is active there. So we know enzyme activity depends on a whole lot of things, like we talked about substrate concentration. We talk right here about temperature. We talked about pH, right? We talked about enzyme concentration. We talked about Kcat, and I always do the K, the lowercase k, as the cursive, just so that y'all know it's not a big K. And then the enzyme Km, right? So all of those things definitely have a really big effect on enzyme activity. So the very last section is the regulation of enzyme activity. And remember that we can have things that are stimulatory and we can have things that are inhibitory, either one. And we typically mediate it by two big mechanisms, bioavailability and then control of catalytic activity. And so I kind of want to show you the difference between the two. We talked about this in class before, where you have your DNA, you turn it into RNA, RNA is processed into proteins. So you are going to adjust bioavailability just depending on how much RNA you make, how you splice it. So that's gene transcription, alternative splicing. Then once you make the enzyme, there are lots of things that you could do to it. You could degrade it and get rid of it. That's protein turnover. You could target it, or you could put it in a vesicle, whatever it is, but that's like sequestration, that kind of thing. But now what we're going to talk about down here, these three down here, are going to regulate catalytic efficiency. So some enzymes, you actually have to cut them in order to make them active. So we call that proteolytic processing. We have covalent modifications where we can add a phosphoryl group or any other group, and we can change its efficiency, or we can bind regulatory molecules to make it work better or slower, either one. But these are protein modifications that will change the catalytic activity of our proteins. So how does this work? Well, typically we talk about enzyme inhibition. And enzyme inhibition is one of two big things. It's either reversible or irreversible. If it's reversible, typically it's non-covalent bonding of small biomolecules. And we can decrease these molecules effects by diluting the enzyme reaction. So if we have a lower concentration of the inhibitor, then it's going to inhibit less, right? And so there are 3 different classes that we'll talk about: competitive, uncompetitive, and mixed, and then irreversible. So irreversible, it's a really, really strong bond. So it's either a covalent bond or lots of non-covalent bonds where it's interacting with the catalytic R groups inside of the enzyme active site, and you're not going to break that bond. And so it kills the enzyme. So as soon as it interacts with an enzyme, that enzyme is done. So even if we dilute the reaction, it doesn't matter. The inhibitor has already interacted with the enzyme and shut down the reaction. So we're going to look at some examples of different kinds of inhibitors. And so I'll give you one example of a reversible inhibitor, and this is succinate dehydrogenase. And succinate dehydrogenase has an inhibitor of malonate. So succinate dehydrogenase's normal substrate is succinate, and it converts succinate into fumarate. But if malonate is present and it has a very, very similar chemical structure, all that's different is we have 2 carbons here where you have 1 there, is that it will compete with succinate to binding to the active site, and it actually has a tighter binding than succinate, and so therefore it will inhibit succinate dehydrogenase. But it's reversible. So if we dilute it, then our malonate will come out and it'll be able to continue to convert succinate to fumarate. So when we talk about reversible inhibitors, there are 3 major categories. We said competitive, uncompetitive, and mixed. So remember that the inhibitor does not, if the inhibitor is bound to the enzyme, then we cannot make product, okay? So in the first one, in competitive inhibition, we have an enzyme. And in that active site, the enzyme either binds to the substrate or it binds to the inhibitor. And so therefore, the inhibitor can only bind to free enzyme, and it inhibits the substrate binding at that active site. So if the inhibitor is bound, substrate can't, no product formed. Uncompetitive means that this inhibitor can bind, but it doesn't bind in the active site. It binds someplace else and induces some kind of conformational change, which prevents the enzyme from converting that substrate to the product. So the substrate will still bind, but it won't make product. So in this case, in an uncompetitive inhibition, the inhibitor only binds to the enzyme substrate complex. It won't bind to the free enzyme. It just doesn't fit. Then we have mixed inhibition. So mixed inhibition is a combination of these 2. And so in mixed inhibition, the inhibitor binds to both the enzyme and the substrate complex. So here we're showing that you could have a free enzyme, and the free enzyme can pick up the inhibitor. That's great. And it can prevent anything from happening. Or your substrate can bind to the enzyme. And if it binds to the inhibitor, then it changes the configuration of the active site. And even though the substrate is bound, it does not make product. The only way you make product is if that free enzyme binds to the substrate, catalyzes the reaction, you get product. So we're going to use our Michaelis-Menten information that we used before, and we are going to explain how these things are going to change our Km and our Vmax. So we know that in competitive, we're going to take each one individually. In competitive inhibition, we have our molecule that inhibits the substrate binding at the active site. And so usually it's an obstruction where if one binds then the other can't, but sometimes it can be a distant site, but typically it's at the active site. And we know that we can test this by increasing our substrate concentration. And if our substrate concentration goes up, the more substrate's there, the more enzyme will bind the substrate rather than the inhibitor. And we can overcome the inhibition that way. So what we expect to see is that our Vmax is unchanged, but our Km values change. And so this is just, you don't have to worry about the equations. What I want you to worry about is being able to interpret a graph. So let's see if I can move this. There we go. Okay. So I'm going to give you the information on the front end, and then we're going to explain it. So in competitive inhibition, we have an increase in our binding affinity, decrease in binding affinity, which is an increase in our Michaelis-Menten constant. And then our Vmax is unchanged. So if we look at the normal graph and we graph concentration of substrate versus velocity of our reaction, what we see is here, the orange is no inhibitor. And as we add more inhibitor, what's happening is it's a much less steep slope. And so eventually this red line, if we kept going and kept going and kept going and kept going, eventually we would get really, really close to the Vmax, just like the orange line is getting. But it would take a really long time. So the Vmax is the same for all of them. And if I take 1/2 of Vmax, so we're at 45, so 22.5. At no inhibitor, that's close to 50 micromolar. At low inhibitor, almost 150. High inhibitor, that's 250. So as we increase inhibitor, what we see is our Vmax changes. But sometimes that's hard to see. I know that it's taking me more substrate in order to reach the same maximum velocity, right? And so if we turn this into the Lineweaver-Burke plot, so the linear version, it's a little bit easier to see. And so what we see is that everything is crossing the y-axis at the same place. Well, the y-axis is Vmax. So you just have to remember which axis is which. Here's my Vmax. They're all the same. So Vmax is unchanged. But now look down here, it's Km. And notice that this is Km apparent, because if you add an inhibitor, you actually haven't changed the binding affinity of the enzyme for the substrate, but it looks that way. That's why we call it the Km apparent. So if we look here, here's no inhibitor. And as we add more inhibitor, it gets closer and closer to the origin. If we go towards the origin, that's telling us that our value is increasing. So our Km gets bigger. As the Km gets bigger, that means that our binding affinity, our apparent binding affinity, gets lower. So you just have to keep practicing this. So example of this is a protease, HIV protease inhibitor that we use, and it typically cleaves at phenylalanine and proline sequences. So it has to be one after the other. So this is our normal substrate structure. And so the enzyme is required for maturation of our viral proteins, but of HIV's viral proteins. And so if we give it an inhibitor, and there are two different inhibitors here, but they have very similar structures to the natural substrate, we can inhibit the viral replication cycle. So uncompetitive inhibition, remember, this is where our inhibitor will bind to the enzyme substrate complex only, alters the conformation of the active site, and therefore you can't make product. So if we look at this, what I see on my Lineweaver-Burke plot is that both the Vmax and the Km are altered. So if you want to figure out how, start at no inhibitor and go to high. So we're going away from the origin. Away from the origin means our value gets lower. So this is our Vmax. So our Vmax gets lower. And also our Km, if we look at that, this is going from no inhibitor to high inhibitor away from the origin. So our value gets lower. So both Km and Vmax are decreased. And what's really important is this is not a competitive inhibition. It's uncompetitive. So you cannot overcome this effect by increasing our substrate concentration. It won't happen. So that's how you can kind of experimentally tell the difference. Mixed inhibition, remember, mixture of competitive and uncompetitive, and it binds to sites that are not near the active site. And it can lower the Vmax, and then you can have one of two things happen. Either it's going to increase or decrease the Km. But there's also something important to remember, that there's a special case of mixed inhibition called non-competitive inhibition. And I'm going to talk about what that is, and then we'll kind of come back to this. Okay, so what we're looking at here is another Lineweaver-Burke plot. And so these are all examples of mixed inhibition. So if we look at kind of the two differences in mixed inhibition, You look at this K1 prime and K1. So this is the affinity of the inhibitor for the enzyme substrate, or the affinity for just the enzymes. This is the rate of association. It's the rate of the reaction. That's why it's a big K. So if it's saying which rate is bigger, has a bigger effect on it. And so for A, that's the enzyme substrate. So if we look here at our intersect with the y-axis, we see our Vmax. So we're going from no inhibitor to high inhibitor. We're going away from the origin. Our value decreases, right? So we have a decrease in Vmax. Here, though, start at the no inhibitor, go towards the high inhibitor. We are going towards the origin, so that's a value increase. So we have an decrease in Vmax and increase in Km. And that's because our inhibitor has a higher affinity for the enzyme substrate complex. In the opposite, right? And I left my arrows on here, but we're going from no inhibitor to high inhibitor. Here we see the similar sort of thing where we see that decrease in our Vmax because we're going away from the origin. But here, our intersection with the x-axis, we're going from no inhibitor to high inhibitor. Ooh, we're going away from the origin. So this is a decrease in Km. So here we have a decrease in both, and that's because the inhibitor has a higher affinity for the free enzyme. And then in non-competitive inhibition, you have a decrease in Vmax where your Km is unchanged. So here's your Km hitting the same place on the x-axis, so it's unchanged. Here we go from no to high. We're going away from the origin. That's a decrease in value. So our Vmax goes down. And it's non-competitive because it has equal rates of reactions with the enzyme substrate complex versus the free enzyme. And that's just what we wrote up here. So my suggestion to you is to try to create something like this for yourself, where you're able to say, okay, what is each of the different kinds of inhibition? What do the Lineweaver-Burke plots look like? And kind of why. So you can try to make that for yourself. So the last set of inhibitors are our irreversible inhibitors. And so this is an example of one that blocks a protease and a phospholipase enzyme. And so when we look at these, this is actually going to form a covalent interaction. So right in here with our serine 195, we're going to lock in this inhibitor in the active site. And when that is locked in the active site, you are not going to have anything else bound to it. It's done. It's stuck inside of it. And so that's the other reason why we sometimes call these irreversible inhibitors suicide inhibitors, right? Because once they react, they're irreversibly bound to the enzyme and nothing else can happen. And so it's usually only happens with a very, very specific inhibitor enzyme interaction. There's some inhibitors that bind to many, many different enzymes and some that bind very specifically. And suicide inhibitors tend to be one of them. So when we talk about other ways that we inhibit reactions, allosteric regulation is a big one. So in this case, we have this enzyme called ATC case, or aspartate trans carbamylase. And this is an enzyme that's required for the production of CTP. So here's the enzyme. It's the pink and the orange, pink and yellow, I guess. The pink is a regulatory subunit of the protein, and the yellow is the catalytic subunit. That's what does the enzyme activity. And so this regulatory subunit can be bound to two things. It can be bound to CTP or it can be bound to ATP. If it's bound to CTP, then it's inactive. And that kind of makes sense. This happens in lots of biosynthesis pathways, is that the final product, the CTPs, the final product, is an inhibitor of usually the first step and sometimes multiple steps. But if CTP is low, it's not going to bind, and we have enough energy in the cell, and we have enough ATP, then this complex becomes active and converts carbamyl phosphate and aspartate and turns it into N-carbamyl L aspartate, and that's the product. And so this is a very early reaction, but both of these two molecules, CTP and ATP, are acting as allosteric regulators. CTP is a negative regulator, ATP is a positive regulator. And it makes sense. Typically, our end product is the negative regulator. And then ATP, there are two reasons why ATP is a positive regulator. Remember, you want an equal amount of purines and pyrimidines. So that ensures that that is equal. And also ATP is energy charge. So if your energy charge is low, you're not going to be making any of the nucleotides because you're not going to be DNA replicating, right? And you can look at this really cool animation of that regulation. And so oftentimes when we talk about allosteric regulation, it's always these either end products or intermediates that come back and inhibit parts of the pathway. So typically it is the end product that inhibits the first enzyme in the pathway, which is what we talked about in ATC ACEs case. But not always. It's not always the first one, but a lot of times it is. And so this is just exactly the same diagram, just looking at it in a different way. This is carbamyl phosphate, this is L-aspartate, and then ATC case will convert it to N-carbamyl aspartate, and eventually that becomes CTP. So our CTP, our final product, comes back and inhibits that very first enzyme in the pathway. And so if you look at the same enzyme and we just change some reaction conditions, let's say we add more ATP, well, then our enzyme becomes more active. If we add CTP, that's the red, we're going to inhibit its activity. And that makes perfect sense, right? Okay, the last kind of thing to talk about is covalent modifications. And we've talked about this before, where it can be phosphoryl groups, methyl groups, acetyl groups, adenyl groups, uridyl groups, all kinds of different things. But the most common is the phosphorylation, this is phosphate group, of serine, threonine, and tyrosine residues by kinases. And remember, kinases are the ones that add that phosphate group to it, whereas phosphatases remove the phosphate group. And so we'll just use one quick example of this with glycogen phosphorylase. And glycogen phosphorylase is something that degrades glycogen into glucose. So it goes glycogen to glucose for energy. And so if you have glycogen phosphorylase and it is not phosphorylated, that in this case is an inactive form. And so in order to become active, it has to go through phosphorylase kinase to be phosphorylated, but that only happens if you're giving off glucagon or epinephrine. So you haven't eaten in a while, or there's a bear that came in the back of the classroom and you're scared and your heart races and you need more sugar in your bloodstream. Those are turning it into this active state. Now, if you just ate and you have high blood sugar, you want to turn off glycogen phosphorylase. So glycogen is stored instead of broken down. And so it's just how we can do these molecular on-off switches. The other way that we can change an enzyme's activity is through proteolysis. Pepsinogen is an example of that, where pepsinogen actually starts off as this blue and it turns into the yellow. This is the N-terminal portion here. And it actually blocks the active site. Here's the active site, and there's already something in the active site. And so in order for pepsinogen to become active, it has to cleave that off. But that only occurs when you hit low pHs. Then you have a cleavage event that gets released, and now we can have something binding in that active site. And so this is another enzyme. This is chymotrypsinogen. And this one actually has to have 4 different cleavage events. So the first cleavage event, this is the full length protein. The first thing is it's cut by trypsin. That's why we call it chymotrypsinogen, because it's cut by trypsin. And then it's also cut by chymotrypsin 3 different places. So that's why we call it chymotrypsinogen. And those cleavage events eventually lead to an active protease. And that is it. Hope y'all have a great day.